Well, I'm going to start by making a confession and admit that I feel a bit like the guy in the couch there, being a CRISPR novice standing in front of a bunch of CRISPR experts. But my talk is going to take a slightly different tack, and I'm going to be discussing the problems and pitfalls that we've encountered in our first attempts at using the technique. So the protein that we're interested in is our lysine deacetylase enzymes, KDACs, also known as histone deacetylases, or HDACs. And the function of these proteins is to regulate the acetylation status of cells. So acetylation is a post-translational modification in which an acetyl group is added to a lysine residue through the actions of lysine acetyl transferase. And this acetylation removes the positive charge from lysine, which results in a conformational change and a functional effect. So KDACs catalyze the reverse reaction, and the level of acetylation is dependent on the balance of actions of CATs and KDACs. A decrease in KDAC activity would result in an increase in the acetylated environment in the cell. So most KDACs are present in the nucleus, where their function is to regulate histone acetylation. Histones interacting with DNA and are therefore responsible for gene transcription regulation. So what is the function of the non-nuclear KDACs? Well, they are involved in regulating acetylation of cytosolic proteins. In our lab, we're most interested in smooth muscle, and in smooth muscle, KDAC8 is present in the cytosol, as you can see here in red, and this is in contrast to KDAC1, which is present in the nucleus, seen here in yellow. So acetylation of non-nuclear smooth muscle proteins is thought to play a role in contractile function, and our lab has shown this using the specific KDAC8 inhibitor, compound 2, in uterine tissue. You can see that in the presence of compound 2, there's a reduction in contractile function. So the proteins that are normally deacetylated by KDAC8 are somehow playing a role in reducing contractile activity. So my project is interested in learning more about how acetylation regulates contractile function. And the specific role of this bit of work was to selectively knock out KDAC1 and KDAC1 in the nucleus and KDAC8 in the cytosol using CRISPR-Cas9 gene editing in order to assess the implications of removing these enzymes on protein acetylation and cell function. So our initial experiments attempted to knock out KDAC8 in human myometrial cells. Primary smooth muscle cell cultures were prepared from uterine biopsies obtained from term pregnant women after written informed consent. Now, our lab has a little bit of molecular biology experience, but we didn't have the resources, time, or manpower to make our own KDAC8 knockout plasmids. So we thought that the best chance of success was to obtain these commercially, which we did from Santa Cruz Biotechnology. And these knockouts consisted of a pool of 3 plasmids, which each had the Cas9 nuclease and a GFP for visual verification. These cells were co-transfected with an HDR plasmid to avoid non-homologous end joining and ensure precise gene editing at the double-stranded break. And these plasmids consisted of an RFP reporter gene and a puromycin-resistant gene for selection. So post-transfection, we found that a few of the cells had picked up the CRISPR knockout plasmid, shown by the positive GFP signal. And of these, some had also picked up the HDR plasmid. And we were quite happy with the transfection efficiency, actually, considering these were primary human cell cultures. After 2 days, there was not really an increase in transfection efficiency, and the morphology of the cells had changed and they were starting to die off. So we moved on to puromycin selection. So the cells that had picked up the CRISPR knockout had HDR, and so they would be GFP positive, RFP positive, and puromycin resistant. And we found that cells were surviving that had all these three qualities, but 4 days after puromycin, we had very few cells remaining. The ones that had only picked up the CRISPR knockout plasmid had died off, and only the ones that had HDR plasmid were surviving, but these cells were few and far between, and they were really spaced out, and we tried to culture them further and they just died off. So after 3 attempts, we decided we better move on to something different. So we went down the commercial route again, and we obtained cells that had KDAC1 and KDAC8 already knocked out with CRISPR Cas9 gene editing. These HAP1 cells are a human model cell line from Horizon Discovery. So we obtained the wild type and 3 knockouts of each of the proteins, each one having a different frameshift mutation, which was initially confirmed by Sanger sequencing. And we looked for the localization of KDAC1 and KDAC8 and found KDAC1 was present in the nucleus and KDAC8 predominantly in the cytosol, as was the case in smooth muscle cells. So because we were looking at selective knockout of the KDACs, we wanted to make sure that the protein was completely absent in the cells. And the obvious way to do this is by using Western blotting. So we looked at KDAC1 first, and we found, as we'd hoped and expected, that the protein was present in the wild type and the KDAC8 knockouts, but not in the KDAC1 knockouts. So this was good, and we moved on to then look at KDAC8. And to our surprise, we found it seemed to still be present in the KDAC8 knockouts. So we tried another antibody, found the same result. Another antibody, found the same result. So at this point, we didn't know what to think. Was the CRISPR gene edit being corrected somehow, or was the protein so stably expressed that it was still present despite the gene editing? So we spoke to Horizon, and they were quite convinced that their knockout was successful. And their local collaborators had mentioned that there was a problem with these KDAC8 antibodies, and they suggested one that they were more confident in. And lo and behold, it did show knockout. So we still needed a bit more convincing. So we went on to look at the protein levels using liquid chromatography, tandem mass spectrometry. So we took lysates of the wild type and the 3 KDAC8 knockouts and separately incubated them with the antibody that showed KDAC8 knockout. And we immunoprecipitated with this antibody, and we ran the precipitates from wild type and the 3 knockouts on a gel to separate the proteins by size. We cut the gel at around 42 kilodaltons, where we'd expect to find KDAC8 protein. These proteins were trypsin digested and then run on the LC-MS machine. And from this, we detected unique KDAC8 derived peptides in the wild type, but in none of the knockout clones. So now we were happy that the protein had indeed been knocked down. So what have we learned from our CRISPR experience? First of all, going down the commercial route doesn't always make things easier and more successful. And it didn't actually fast track the process of answering our biological questions. Nothing is ever that simple. It seems CRISPR-Cas9 may be less amenable to use in certain cell types, as we found with the uterine smooth muscle cells. This might be because of low transfection efficiency, or it could be the cells grew too slowly, or it could be the effects of KDAC8 but knockout, but we never got far enough to get enough material to test that out. And we came across the age-old problem of the non-specific antibody, which can make verification of knockout either straightforward, as was the case with KDAC1, or it may become troublesome, time-consuming and expensive, as was the case with KDAC8. It's only now that we've been able to examine the functional impact on the cells of increasing acetylation levels and how this is influenced by removal of KDAC1 from the nucleus and KDAC8 from the cytosol. And we've performed some microarrays to look at gene expression in all the different clones. And And to our surprise, there doesn't seem to be any difference in gene expression, even when KDAC1 was knocked out, either at baseline or in an increased acetylated environment following TSA treatment. And this suggests that there's a compensatory effect of the other nuclear KDACs covering for the absence of KDAC1. So thank you for listening, and I'd like to thank Paweł Pomolski, who did the mass spec, and our funders for supporting the work. Thank you.